Good day everyone, I'm David, moderator for this webinar. I welcome everyone for today's webinar, which is Sterile Filtration of Pharmaceutical Products, Validation and Regulatory Requirements. I would now like to introduce our presenter for today, Roger Cohen. Roger Cohen is the founder and owner of RC Cohen Consulting Services LLC. He has 35 years of experience in pharmaceutical quality assurance and manufacturing. I would now like to hand over the floor to the speaker. Over to you, Roger.
Hello, my name is Roger Cowen. I'm here today to present to you a webinar entitled Sterile Filtration of Pharmaceutical Products. Perhaps more importantly, what you need to know to meet validation and regulatory requirements. Now, I'm going to start off with a little description of the quality rationale behind sterile filtration. First off, it is one of the most critical steps in the manufacture of a sterile pharmaceutical product. What are some of the applications behind sterile filtration? Now, a little history behind sterile filtration here. Now, sterile filtration using size exclusion was discovered to have the benefit of removing bioburden with minimal effects for liquid or gas properties. Let's look at some of the devices that are out there for sterile filtration. The one that's most commonly used in pharmaceuticals is the stainless steel filter housing. Also, another device that's increasingly being used are the plastic disposable capsules. Another device are the disc filter holders. Okay, so those are some of the devices that are available. How do you select what type of sterile filter for your application? The other another characteristic is retention. Another characteristic for your sterile filter selection is flow rate. Now, capacity is slightly different. Capacity of a filter is important. Another characteristic when looking for a sterile filter is wettability. Now, hydrophilic filters and hydrophobic filters are two types of filters that are differentiated by their wettability. Okay, we're going to dive a little deeper here and actually look at the structure of the sterile filter membrane. The sterile filter structure, there are two types. There's symmetric membranes and asymmetric membranes. Now, symmetric membranes have a uniform structure throughout their depth. Asymmetric membranes, on the other hand, the structure varies through a depth cross-section. Now, some of these polymers that are available, there's PVDF, that stands for polyvinylidene fluoride. These are hydrophilic filters. Polymer is PES, polyether sulfone. These are hydrophilic filters. You can see a photomicrograph to the right. Another type of polymer is nylon 66. It's hydrophilic, polyultrapure 66. This exhibits low extractables, low protein binding. Cellulose ester membrane filter is the one pictured to the right. This is used for microbial analysis purposes. And this is the subject of redundant filtration or serial filtration. Now, this refers to the use of two sterilization filters in series. Now, there are three terms describing this. There's redundant filtration. That's a term that's used when one of the filters is used as a backup for the other when one fails post-integrity testing. Now, when you're deciding whether to use redundant filtration or not, it's a good idea to go over the risk factors. How about sterilization of filters? Well, there's three major methods used to sterilize filters. Gamma radiation, autoclave, and steam in place. Steam in place is a method that's most effective with assembled stainless steel systems. When you're validating a sterile filter system, there's five major considerations: filter inertness, compatibility, absorption, extractables, but also microbial retention capability and integrity. First off, inertness. So the septic filter must be validated as inert. So one part of inertness is compatibility. Now, another measure of filter inertness is adsorption, if the safety or efficacy of the drug is negatively affected. Okay, so another measure of filter inertness is extractables. Again, measuring extractables, you can do a couple of weight type measures. First one is gravimetric extractables. Another similar test is called weight change. Now, some of the analytical procedures that are used for extractables are high performance liquid chromatography, HPLC, and Fourier transform infrared spectroscopy. Another analytical procedure that's used for extractables is TOC, or total oxidizable carbon. Another extractable that often gets forgotten about is particulate. Now, particulates in a finished pharmaceutical, it may also have originated from non-filtered sources. Another potential extractable is endotoxin. How about toxicity? A thorough validation study is needed to determine if filtration of the drug product produces any tox effect. How about bacterial retention testing? And the chosen model organism or test organism that's used. Now, custom retention testing is endorsed by the FDA. The Sterile Processing Guideline 2004 I've got pictured to the right the typical apparatus used for bacterial retention testing. The viability of the test organism in the drug product under process conditions is also assessed. How about responsibility for this? Well, from a regulatory perspective, it is the responsibility of the pharmaceutical manufacturer. Okay, different type of validation, integrity testing. It is the responsibility of the filter manufacturer to establish some minimum integrity test specs. Two types of filters, as we said, hydrophilic and hydrophobic. The main one that's used is bubble point integrity testing. It's most commonly used. It's non-destructive. The second method is diffusion integrity test. Another similar test is a pressure hold integrity test. Now, I referred to this one earlier for hydrophobic filters, the water intrusion integrity test. Okay, so when you're performing integrity testing, something called filter flushing becomes very important. Now, what are some of the challenges behind filtration? One of the big challenges I've seen is high viscosity. Another challenge is extended use of your sterilizing filter. Now, some other challenges are specific types of organisms. Spirochetes on responsibility, filter user versus filter manufacturer. The filter manufacturer can provide retention claims and provide test methods. So that brings us to the end of the webinar.
Okay, thank you, Roger.
Yes, I realized that this webinar had an awful lot of detail in it, and I apologize for that, but I think given the nature of the subject matter, it was needed. And I hope each and one of you has got some little nugget of information out of here that you can use in your own facility. So thank you very much for listening.
Okay, thank you, Roger. That does conclude the session for today. Thank you, everyone, for participating. You may all disconnect now. Have a great day ahead.
Bye now.